Welcome to Study Session 3, The Cell. Introduction. Life starts from somewhere, and a lot of complex processes are involved in the formation or bringing a life into existence. In this study session, you shall focus our attention on the cell as the smallest unit of life. Learning outcomes. At the end of this study session, you should be able to 1. Explain what a cell is all about. 2. Discuss how the division of cells leads to living entities. What is a cell? Cell is the smallest unit of life that can carry out all the functions of a living thing. Cells are the basic units of all life. All organisms are made of one or more cells, and all cells arise from existing cells. Cells are classified into 2 categories: simple non-nucleated prokaryotic cells and complex nucleated eukaryotic cells. These 2 classes of cells do not divide in the same way. Prokaryotic cells were the first form of life on Earth, characterized by having vital biological processes involving cell signaling and being self-sustaining. They are simpler and smaller than eukaryotic cells and lack membrane-bound organelles such as the nucleus. Prokaryotes include 2 of the domains of life, bacteria and archaea. The DNA of prokaryotic cell consists of a single chromosome that is in direct contact with the cytoplasm. The nuclear region in the cytoplasm is called the nucleus. Most prokaryotes are the smallest of all organisms, ranging from 0.5 to 2.0 in diameter. Plants, animals, fungi, slime molds, protozoa, and algae are all eukaryotic. These cells are about 15 times wider than a typical prokaryote and can be as much as 1,000 times greater in volume. The main distinguishing feature of eukaryotes and compared to prokaryotes is compartmentalization, the presence of membrane-bound organelles, compartments in which specific metabolic activities take place. Most important among these is a cell nucleus, an organelle that houses the cell's DNA. This nucleus gives the eukaryote its name, which means the true kernel, nucleus. The eukaryotic DNA is organized in one or more linear molecules called chromosomes, which are associated with histone proteins. All chromosomal DNA is stored in a cell nucleus, separated from the cytoplasm by a membrane. Some eukaryotic organelles, such as mitochondria, also contain some DNA. Many eukaryotic cells are ciliated with primary cilia. Primary cilia play important roles in chemosensation, mechanosensation, and thermosensation. Cilia may thus be viewed as a sensory cellular antenna that coordinates a large number of cellular signaling pathways, sometimes coupling the signaling to ciliary motility or alternatively to cell division and differentiation. The plasma membrane resembles that of prokaryotes in function, with minor differences in the setup. Cell walls may or may not be present. Cell structure and function. A typical cell has an outer non-living layer called cell wall. The cell membrane is present below the cell wall. The cell membrane encloses protoplasm. The protoplasm has a semi-fluid matrix called cytoplasm and a large membrane-bound structure called nucleus. The cytoplasm has many membrane-bound structures like endoplasmic reticulum, Golgi bodies, mitochondria, plastids, microbodies, vacuoles, and non-membranous structures like centrosome and ribosomes. These are called cell organelles. The cytoplasm without these cells organelles is called cytosol. The cytoplasm also contains non-living inclusions called ergastic substances and cytoskeleton, microfilaments and microtubules. The content of the cell within cell wall is called protoplast. The cytoplasm without living cell organelles is called cytosol. Comparison of plant and animal cell. In plant cell, cell wall is present. In animal cell wall is absent. In plant cell, centrioles are absent. In animal cells, centrioles are present. In plant cell, plastids are present. In animal cells, plastids are absent. In plant cell, they have large vacuole. In animal cells, they have small vacuoles. Description of the cell contents. 1. Cell wall. It is an outer non-living rigid layer of cell. It is present in bacterial cells, fungal cells, and plant cells. It is a permeable membrane chiefly composed of cellulose. It gives rigidity, mechanical support, and protection to the cell. 2. Protoplast. It includes cell membrane and protoplasm. Cell membrane or plasma membrane. It is a semi-permeable membrane present in all cells. It is present below the cell wall in plant cell and outermost membrane in animal cell. It is composed of phospholipids, proteins, carbohydrates, and cholesterol. It allows the outward and inward movement of molecules across it. The movement of molecules across the plasma membrane takes place by diffusion, osmosis, active transport. Protoplasm. It is a living substance of the cell that possesses all vital products made up of organic and inorganic molecules. It includes cytoplasm and nucleus. Cytoplasm. It is the jelly-like semi-fluid matrix present between the cell membrane and nuclear membrane. It has various living cell inclusions called cell organelles and non-living cell inclusions called ergastic substances and cytoskeletal elements. The cytoplasm without cell organelles is called cytosol. Golgi bodies. Golgi complex has a group of curved flattened plates like compartments called cisternae. They stacked one above the other like pancakes. The cisternae produce a network of tubules from the periphery. These tubules end in spherical enzyme-filled vesicles. Their functions. They pack enzymes, proteins, carbohydrates, and so on in their vesicles, hence called packaging centers. They produce lysosomes. They secrete various enzymes, hormones, and cell wall material. They help in phragmoplast formation. Mitochondria or chondriosome. Mitochondrion is a spherical or rod-shaped cell organelle. It has two membranes. The outer membrane is smooth. The inner membrane produces finger-like infolding called cristae. The inner membrane has stopped particles called Rackus particles. The mitochondrial cavity is filled with a homogeneous granular mitochondrial matrix. The matrix has circular mitochondrial DNA, RNA, 70 ribosomes, proteins, enzymes, and lipids. Common name: powerhouses of the cell, storage batteries of the cell. Their functions: mitochondria synthesize and store the energy-rich molecules ATP, adenosine triphosphate, during aerobic respiration, so they are called powerhouses of the cell. Chloroplasts. These are green-colored plastids containing chlorophylls and carotenoids, carotenes, and xanthophyll. Chloroplast is double membranous cell organelle. The matrix is called stroma. The stroma has many membranous sacs called thylakoids. They are arranged one above the other like a pile of coins to form granum. These membranous structures have photosynthetic pigments like chlorophylls, carotenes, and Xanthophyll. Cell division. Cell division is a process by which a cell, called the parent cell, divides into two or more cells called daughter cells. Cell division is usually a small segment of a larger cell cycle. This type of cell division is known as mitosis, and it leaves the daughter cell capable of further division. In another type of cell division present only in eukaryotes, called meiosis, a cell is permanently transformed into a gamete, and it cannot divide again until fertilization. In eukaryotes, there are two distinct types of cell division: a vegetative division, whereby each daughter cell is genetically identical to the parent cell, mitosis, and a reductive cell division, whereby the number of chromosomes in the daughter cells is reduced by half to produce haploid gametes, meiosis. The growth and division of cells, mitosis, and the formation of sperm and eggs, meiosis, are important for all organisms, including humans. We all began as a single cell that resulted from our father's sperm fertilizing our mother's egg. This single cell then divided by mitosis into two cells. Daughter cells then continued again until we were grown up. It is still happening right now, as some of our cells use mitosis to replace old dead cells, such as skin cells. Meiosis, on the other hand, is a special form of mitosis that occurs only in a special subset of our cells to form eggs and sperm. Mitosis. In mitosis, a single cell divides to form two daughter cells. These cells grow and then divide to form a total of 4 cells. While in meiosis, a single cell divides twice, resulting in 4 daughter cells that do not grow and divide again. Instead, these cells are modified to become eggs or sperm in humans. Mitosis is a part of the cell cycle in which chromosomes in a cell nucleus are separated into 2 identical sets of chromosomes, and each set ends up in its own nucleus. In general, mitosis, division of the nucleus, is often accompanied or followed by cytokinesis, which divides the cytoplasm, organelles, and cell membrane into 2 new cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the meiotic phase of an animal cell cycle, the division of the mother cell into 2 daughter cells, genetically identical to each other and to their parent cell. The process of mitosis is divided into stages corresponding to the completion of one set of activities and the start of the next. These stages are prophase, prometaphase, metaphase, anaphase, and telophase. During mitosis, the chromosomes, which have already duplicated, condense and attach to spindle fibers that pull one copy of each chromosome to opposite sides of the cell. Meiosis. Meiosis results in 4 haploid daughter cells by undergoing 1 round of DNA replication followed by 2 divisions. Homologous chromosomes are separated in the first division, and sister chromatids are separated in the second division. Both of these cell division cycles are used in sexually reproducing organisms at some point in their life cycle, and both are believed to be present in the last eukaryotic common ancestor. In meiosis, DNA replication is followed by 2 rounds of cell division to produce 4 potential daughter cells, each with half the number of chromosomes as the original parent cell. The 2 meiotic divisions are known as meiosis I and meiosis II. Before meiosis begins, during S phase of the cell cycle, the DNA of each chromosome is replicated so that it consists of 2 identical sister chromatids, which remain held together through sister chromatid cohesion. The S phase can be referred to as pre-meiotic S phase or meiotic S phase. In some cases, all four of the meiotic products form gametes such as sperm, spores, or pollen. In female animals, 3 of the 4 meiotic products are typically eliminated by extrusion into polar bodies, and only one cell develops to produce an ovum. Prokaryotes also undergo a vegetative cell division known as binary fission, where their genetic material is segregated equally into 2 daughter cells. All cell divisions, regardless of organism, are preceded by a single round of DNA replication. One of the most important requirements of a successful mitosis or meiosis is that the blueprint of life, the DNA, is provided in equal amounts to each of the daughter cells. The DNA in all human cells except eggs and sperm is housed in 46 chromosomes. Eggs and sperm consist of only half of that number, 23 chromosomes per cell. Every person inherits one set of chromosomes from their mother in the egg and the other set of chromosomes from their father in the sperm. It is imperative that when mitosis and meiosis are complete, the appropriate number of chromosomes exist in each cell. If there are extra or missing chromosomes, the cell usually do not live. Fission is the division of a cell or body, population, or species into 2 or more parts, and the regeneration of those parts into separate cells, bodies, populations, or species. Binary fission produces 2 separate cells, populations, species, and so on, whereas multiple fission produces more than 2 cells, populations, and species. Definition of terms. DNA, deoxyribonucleic acid, is the hereditary material of life and molecules of the nucleus that direct all chemical reactions, metabolism that go on in the cell. Mitosis is the growth and division of cells. Meiosis is the formation of sperm and eggs. Summary of study session. At the end of this study session, you have learned that 1. The cell is of great significance to living entities. We stated that cells are the basic units of life and all organisms are made of 1 or more cells. Cells are classified into 2 categories. 2. Cell division is a process by which a cell called the parent cell divides into 2 or more cells called daughter cells. We have come to the end of study session 3. Thank you.